Hello everyone, my name is Asisipho and welcome to my YouTube channel. In today's video guys, we'll be looking at DNA replication and transcription, basically describing the two processes and highlighting the important points that you need to mention in order for you to get the full 6 marks when you're asked to describe the processes. Now, learners tend to confuse the two processes, so I'm going to describe the points for each process and address common mistakes. So please do watch till the end guys, so that you understand some of the mistakes that learners make and you obviously try to avoid making the same mistakes. So without wasting time, let us look at the points for DNA replication. Now, we already know that the DNA double helix unwinds. DNA double helix unwinds. That's the first point. And the hydrogen bonds break. Then you have hydrogen bonds break. Whether you write the word weak or not, it doesn't matter. What is important is noting that the hydrogen bonds break, or you can say the double-stranded molecule unzips. For this particular point, double-stranded molecule unzips. Then for the third point, you need to mention that we are now going to have two separate strands to form two separate strands. From those two strands, under DNA replication, you can either say both or each strand is going to serve as a template. So both, you don't have to use both words. So you can either choose to use both or each, right? Each strand serves as a template. To build a complementary DNA strand. Remember, we're making a copy, right? DNA replication, we're making a copy of DNA. So to build a complementary DNA strand. And how will that work? We are going to have A pairing with T. So A pairs with T. You can even write the full words. It's also fine. And C is going to pair with G. Using what? How does the pairing work? We're going to be using free DNA nucleotides. And where are these DNA nucleotides? They are in the nucleoplasm. So using free DNA nucleotides from the nucleoplasm, and this will result in two identical DNA molecule. This results in two identical DNA molecules. Now let's talk about the important words and everything that you need to mention in order for you to get full marks. This is very important. And remember guys, if you have not subscribed to my YouTube channel, please kindly hit that subscribe button. Please like the video, comment down below, and share the link with your schoolmates, classmates, and also your friends. All right guys, let's look at how this would be marked, right? I'm going to change, let me use this one. a color. So DNA double helix unwind. So in order for you to get this 1 mark, you need to mention the word DNA and the word double and the word helix and the word unwinds. So that whole point needs to make sense. You need to mention to us that you are talking about the DNA and you describe the shape of DNA and that it is now unwinding. So if you don't mention the word DNA, you won't get a mark. If you don't mention double helix, you also won't get a mark. Now, looking at the second point, there's 2 options for that point. Hydrogen bonds break. You need to obviously mention that these are the hydrogen bonds that are breaking. If you just say bonds are breaking, which ones? You know, if you don't mention that these are hydrogen bonds, you won't get a mark. The word weak doesn't really matter in this case. Whether you mention that it is weak hydrogen bonds or not, it doesn't matter, the word weak. Now, for the second point, if you don't get a mark for hydrogen bonds break, you can get a mark for saying the double-stranded molecule unzips. So you're going to get 1 mark from these 2 options, okay? Then we know that after the hydrogen bonds break, there'll be 2 separate strands that are going to be formed. That's the 3rd mark. Now you need to mention how many strands are formed. There are 2, and they'll be separate because now the bonds in between will no longer be there. Okay. Then this is very important. I can't help but emphasize how this word is important, and the reason for that is because I think the first 3 points are very similar. They're actually the same 2 transcription points which we're going to look at now. But with the 4th point, guys, in transcription it says only 1 strand. In DNA replication, it talks about both strands serving as template, or you can say each strand, since there's 2, each meaning both actually. They're going to serve as a template. So this word is important. So you can either use both or each. There's no need for you to use the 2 of them. Okay. And these strands are going to serve as a template to build a complementary DNA strand. Now, you can get a mark for saying to build a complementary DNA strand, or hence there's a slash here. You talk about the bases that are going to pair with the template strands. So in the nucleoplasm, we do talk about the nucleoplasm here, but it's fine. I'll just talk about A pairing with T and C pairing with G. It doesn't matter which one you start with. You can say G pairs with C, T pairs with A. It doesn't matter. You'll just get a mark for that. So how many marks do you have so far? 1, 2, 3, 4, 5. Okay. Now, the pairing of the bases here is obviously due to the free DNA nucleotides. So now using free DNA nucleotides, and where are we getting these nucleotides? In the nucleoplasm. Common mistake. If you don't mention the word DNA, you actually don't get a mark, because remember, in the nucleoplasm, there's also RNA nucleotides. So you need to mention that. In DNA replication, we're going to be using DNA nucleotides, right? So that means those are nucleotides that are carrying the deoxyribose sugar, all right? Okay, and where are they from? They are from the nucleoplasm and not the cytoplasm. Maybe I need to mention, and not the cytoplasm. Remember, cytoplasm is that jelly-like liquid within the cell. The nucleoplasm is the one within the nucleus. So not cytoplasm. Please don't confuse the 2. Then, the last point, and I'm also going to address common mistakes. This results in 2 identical DNA molecules. Remember, we are making a copy of DNA, and DNA is a molecule. So now that we've formed another one, we are having 2 identical DNA molecules and not 2 identical strands. That is important. So if you're talking about 2 identical strands, that is actually incorrect, because remember, in that molecule, if we were to scroll up, in that molecule, and I don't want to make this video to be way too long, in that molecule we had that double helix molecule, which is the DNA, right? And you've mentioned molecule here, and it unwinds to form 2 separate strands. So the word strands is correct here, because that molecule unwinds to form these 2 strands, and these 2 strands are going to act as template. This will be a template strand, and this will be another template strand. Then we build complementary strands. We're going to build complementary strands here in the middle. There's a complementary strand, there's a complementary strand. Okay, with the bases that are going to pair, right? Then what do we end up having? We end up having two separate, not separate, two identical DNA molecules. So this will be one DNA molecule and another DNA molecule having what? Having a template strand and a complementary strand. Template strand, complementary strand. I hope it makes sense, guys. So these are really some of the common mistakes that learners make. Okay, now let's look at transcription. Straight from exam guidelines, guys. Straight from exam guidelines. Transcription. Let's see. So the DNA double helix unwinds. Yeah, DNA double helix unwinds. All right. Then second point will also be the same. Hydrogen bonds break. Hydrogen bonds break. Or you can talk about the double stranded molecule unzips and zips. I think another thing I forgot to mention, some learners swap these points, which will obviously make you lose marks. So some talk about the DNA molecule unzipping and the weak hydrogen bonds unwinding. Yeah, or even these two words, unzip comes after unwinding. So make a way, just find a way to remember. It unwinds first, then unzips. Okay. Then the third point, to form two separate strands. Let's see. Double-stranded molecule unzips. Yes. To form two separate strands. Can you see that the points, the first 3 points for both DNA replication and transcription are the same? Same. Right? So whether you're describing DNA replication or transcription, the first 3 points are the same. Now the points differ from the 4th point. I think I've mentioned it already. So yes, we're going to have 2 separate strands forming, but from those 2 strands, only one is going to be used as a template. One strand is used as a template. I'm sure you can see the difference. Okay? I'm sure you can see the difference. So in transcription, one strand is used as a template. In DNA replication, both strands are used, right? Or they serve as a template. Okay? But now this one strand that is used as a template or that serves as a template is actually serving as a template to form messenger RNA. To form messenger RNA. Now, how exactly is messenger RNA formed? Messenger RNA is formed by using nucleotides, free nucleotides. But which nucleotides in this case? They are going to use free RNA nucleotides. Then where are we finding these nucleotides? In the nucleoplasm. I'm sure you can see the difference, right? So we had said in DNA replication, we are using free DNA nucleotides. But in in transcription, we are using RNA nucleotides because now we are forming a messenger RNA, which is an RNA molecule. Okay, so that means these nucleotides are obviously carrying the ribose sugar. Okay. The messenger RNA will now be complementary to the DNA. So that's the next point. Messenger RNA is complementary to the DNA. Okay. And now we can say that messenger RNA now has a coded message for protein synthesis. Those are the points. Let's look at the marking for transcription. Okay, so DNA double helix unwinds, I've explained that. So the word DNA double helix unwinds will need to be there. Weak hydrogen bonds break or the molecule unzips to form two separate strands. One strand is used as a template. In DNA replication, both to form messenger RNA using free RNA nucleotides in the nucleoplasm and not cytoplasm. Now, messenger RNA is complementary to DNA, and messenger RNA now has the coded message for protein synthesis. I'm sure you can see, like I've mentioned already, the first 3 points for both DNA replication and transcription are the same. Then the next points obviously will differ. All right. I hope it makes sense. Remember to subscribe to the channel, guys. Hit that subscribe button. Please like the video. Share the link with your classmates and your schoolmates. And all the best for your exam.